Hi everyone, welcome back to a new term. So in this term, we'll be starting off with a quick revision on the cell division. So we'll start off first with cell cycle, a quick broad overview of this topic. And we'll go into DNA replication. For those of you that have done the SLS, you will have come across this particular part of the SLS where there is a video to learn about how DNA replication works. This particular segment may confuse some students, but I try my best to teach as properly as possible in this video. Actually, what I'm going to do today is just a physical live version, but in a slower way. Okay, so a revision on that. After that, we're going to do a quick revision of mitosis, as well as meiosis. And more importantly, how to identify the phases if you were be given microscope images. Okay, so that's probably what we'll be doing today. But maybe on your part, very quickly, before I go through every single segment, I would like you quickly to just scan through, revise very quickly, cell cycle. What are the various phases of the cell cycle? DNA replication. Does this still look familiar to you? Mitosis, meiosis, what are the phases? So that as I revise, then it is a triple revision for you. Some of you for a quick refresher. Okay, we're going to slowly begin. It's okay, we don't need to revise everything yet. We start off first with cell cycle. When we look at the cell cycle, as the name implies, it's a cycle. So what this implies is that we expect that cells over generations will go through this kind of standard model of life. When we look at the cell cycle, there are many phases to the cell cycle, some lasting longer than another. They are often expressed in a pie chart to show you how much time it spends in each phase. Now, what I've drawn here definitely is not to scale, but what relatively speaking is quite to scale is the proportion between these three phases and these smaller phases here. The cell actually spends a lot of time in these three phases. In fact, right now, most of your cells are in these three phases. If it's in these phases over here, you literally, before your very eyes, you'll see your friends dividing on the skin surface, right? Suddenly one new flake of skin come out, right? It's very disgusting. So actually, in truth, our cells are kind of in this phase right now. Okay, perhaps let's break down what this giant phase over here is. What is this big phase over here called? Oh, thanks, Isabel. Okay, Isabel says it's interphase. Yeah. Think of it as like the intermission, the long break before the action happens. So interphase. Interphase can be broken down into three broad phases if we go in this direction. First, we have what phase? Okay, G1. G1 phase. I also used to think that G means growth phase, but actually turns out it means gap. Yeah, so it's like gap 1. In this phase, however, you do expect a lot of growth taking place. The cell is growing, dividing its parts. Then after G1 phase, it goes into the next phase called the, thank you. Okay, then we do together as a class. S phase happens during S phase. Maybe what does S stand for? Synthesis. Synthesize what? DNA. Okay, so from here onwards, you pay attention. We'll always be talking about DNA now from in this term, because that's really what we are going to, what our body is working with. DNA replication. This is when DNA divides. Then we go into the next G2 phase, where a little bit more growth takes place. Here is where things divide. Very broadly speaking, the cell will first undergo something called nuclear division. After which, then it will go through really the division of the cytoplasm or the cell. There's a difference between there's a reason why I separate these two. Because we know that cells come with nucleus. What this means is that, unlike what we may have believed, the cell doesn't just pinch inwards and divide like that into two. It's not like a very easy chop like that. Actually, interestingly, the cell first divides its nucleus from 1 to become 2. After it divides its nucleus, then it divides the cell into 2. So, that's what you've been spending the holidays learning. Mitosis or meiosis. It refers to the division of the nucleus, not the entire cell. However, we call this chapter cell division because after the nucleus divides, then the cytoplasm and the cell divides. Okay? So I just want to make things very clear because sometimes if some of us may say that mitosis is a form of cell division. It's not, it's the division of the nucleus. Before we do a quick revision on these parts, I have a very important question to ask. Why must we divide, or why must we replicate our DNA before we go into nuclear division and cell division? Everyone have a think, huh? It's a key question. Why must we go through this before we do this? What will happen if we do not have this? Okay? Yeah. Maybe I hear from Brown. Red, what do you think? Why do we need to divide our DNA into before we? Why do we need to divide our DNA into two, replicate it before we start to divide the nucleus and the cell? Okay? Yeah. Yeah. So that, okay, we use a term called daughter cells. Yeah. Daughter cells are the cells that arise after you divide. So, yes, we do need this particular phase over here so that we don't end up with daughter cells with less and less and less DNA. Yeah? We need to ensure that we have enough things to pass over to the next generation before we divide. Okay? Alright, so now let's go into mitosis. Mitosis consists of 4 phases. What are the 4 phases? Yeah, I'll go fast. Huh? I think the acronym people always use is PMAP. Okay, so it's Prophase, metaphase, anaphase, telophase. What happens in each phase? That is what you're expected to know. You're also expected to be able to identify the phases if we give you photos. So there are some sample questions in the SLS for you to practice identifying. It's an important skill because you may be looking at the microscope and like, hey, what's this? Or we may give you a structured question and then we show you the photo, like, what stage is this? Last but not least, there is cytokinesis. That is when the cell truly divides into two. And so that's the cell cycle. Broadly speaking, this is what many cells go through. Do all cells go through this? Actually, some cells don't. Some cells don't go through this phase after they have decided that this is the cell I'm going to be for the rest of my life. Yeah? So for example, your brain cells, once they've differentiated from normal cells to become brain cells, you don't really go through these phases anymore. So take care of your brain. Don't anyhow whack it. Yeah? Because it's not so easy to replenish. Some cells go through this phase many times. For example, stem cells. Before you were yet a fully grown baby, the stem cells are constantly dividing. If you were to injure yourself, there are some stem cells that will be able to divide, quickly replace and replenish. Within your bone marrow, you also find a lot of stem cells. They go through this phase a lot. So overall, the cell cycle helps in your growth, replenishing things, creating new cells. Okay, and that is the cell cycle. The next part of the lesson we're going to quickly revise would be the S phase. The S phase is the phase I'd like you to pay more attention to, because truly it's this phase that is very important. As Rin pointed out, if we do not replicate our DNA, we are going to end up with cells with less and less DNA over generations. So it is our role to understand how DNA replication works. However, in truth, I do not need you to know how it works on a mechanism level. Instead, I would like you to appreciate this particular experiment that two brilliant scientists did a long time ago. They are named Mitosis and Starr. I'm not sure if they're still alive, but recently when I went to YouTube, I could find two old fellas talking about their own experiment. So if you go online, type Mitosis and Starr, right? You can find two very cute old gentlemen. Let's just find them now. I don't know, let's go to Lumos. But I don't have So just 2 years ago, I found a video of very individuals that came out with this experiment.
John Kerry sat on the telephone and very excited once he had just read animals. And you know, they are the most beautiful experiments in biology. And I gasped. I said, John, you didn't say that. You said, the Michelson stall experiment was the most beautiful experiment in biology. Oh, did I? Well, I was wrong. Once McCarthy made the discovery, they proposed a model. There are those who believe this model must be true because it was so beautiful. And there are those who believe it must be wrong because biology is complicated and this model is too simple to be right. It is simple, but there was no experimental proof. They have a model. From very early childhood, I mean practically infancy, I love science. I love putting wires together and making little radios, which I could put under my clothes so my parents wouldn't know that I was listening to them all night long. And then I was very, what makes life work. And there has been, I think, in the house, painting in my bedroom, and painting by Michelangelo, the God of Fire, and touching fingers. And I don't know if there is a spark. I don't think there's a spark, but I'm not sure. But to me, that life is somehow electric. God is providing life through a spark. And for some reason, that made me interested in electric things. Unlike that, I have no particular strong interest in science. It was understood that when I graduated from high school, I would apply to the Navy. That's what my mother had in mind because she thought I would be good at just once. But then, of course, World War II broke out, so that kind of changed fast, and she decided I should just go to college. I think I was just too young to understand the forces of the Americans. I hadn't had enough life experience to get a grip on the questions they were really thinking about. Science, on the other hand, was concrete. Children can grasp science. And among the sciences, biology was the most appealing. There, the fun was to figure out puzzles. That is, there was a rational, concrete, quantitative explanation for what you saw. You could reason backwards as to what was going on. And that intrigued me enough to know that genetics was something perhaps I could do.
This is a long video. If you're interested, you can look this up. You can also find Mitosis himself teaching his very own experiment in a lecture. And I think that is quite a different experience when you hear it from the scientists themselves. And I think what I enjoy about this video is that you see that many of them didn't set out to want to discover something new or to do this experiment. And one thing leads to another. Parents decide want to do this, war breaks out. But they are where they are from being open-minded and trying new things. So I think for yourself, you do not need to know how DNA replicates on a mechanism level, but I would like you to appreciate how this experiment helped us understand how DNA replicates. That is your goal. What you may be assessed on would be variations of this experiment. We may show you this same experimental procedure, often in structured questions or in MCQ, and then we will ask you, what happens if we tweak a certain part of the experiment? What do you think the results would be if we tweak the experiment? Okay, so now a quick revision on this particular experiment. If I'm not clear, again you can rewatch the video again. Okay, so in this morning, I'm going to share with you the most beautiful experiment in biology, the Mitosis and Star experiment. This experiment is most beautiful, you'll see in a while, because it's very elegant. It's not complicated at all, very simple. Here it helps to solve one of life's most fundamental questions. But what is that fundamental question? Actually, in the past, people did not know how DNA replicates. They know for a fact that DNA comes in pairs, correct? A DNA molecule we know of is double-stranded. Yeah, that's one of the properties of DNA. So if I were to show you DNA again, today I'm going to represent all the DNA double-stranded just with two thick bars. Okay? So for example, if I were to look at DNA, I'm going to write down original and original as so that you can keep track of where this DNA is going to be later on. If we look at DNA, it comes in two strands. One strand, two strand, double-stranded. That's what I'm going to show you today. But actually scientists did not know how DNA, a double-stranded DNA, replicates. Because if you think about it, there are a few possibilities. The first possibility is that perhaps if I have a double-stranded DNA, maybe the first logical thing would be this. Let's create new DNA. Okay? So I'm going to represent any new DNA with this word new. You can see at the back screen. Okay? So supposing I want to create new DNA. maybe this is what happened. So this was theory number 1, or hypothesis number 1. We take the original strand of DNA. Okay, so this is perhaps generation 1, this is generation 2. Let's take the original strand of DNA. Next, first thing we do, separate them apart. And we create new DNA. Now the first theory goes is that after this new DNA has been created from the original strand, the 2 new strands will then join together. The original strands will rejoin. And this is what we'll be left with. We'll be left with the original DNA left intact, as well as new DNA molecule, double-stranded, totally made from new nucleotides surrounding them. We call this theory, this hypothesis, the conservative model of DNA replication. Conservative because the original double-stranded DNA is still present. Yeah? Perhaps it makes a little bit more sense if we look at it from a semi-conservative mode. Semi-conservative model. Because in the semi-conservative model, well, we start off with an original strand, and as we predict, likely we have to separate the strands before we can create new strands. This particular model predicts that, actually it's kind of troublesome to separate the new strands away from the original strands and to rejoin to each other. Why not just stay that way? After a new strand has been synthesized from the original strand as a template, the new strand has been synthesized from this original strand as a template, let's leave them together. Now I'm left with 2 DNA molecules, half original, half totally new strand. And that's why we say semi-conservative, because it's only, each DNA molecule is only semi-conserved. There's only one original. Whereas in over here, we have fully conserved. Those are the 2 hypotheses of the time. What is beautiful about Mitosis and Styles' experiment was that it helped to scientists realize which model is actually true, which one is actually happening on a microscopic level in your cells. And I think one of the most beautiful things that they applied was from chemistry. Because chemistry, there are isotopes, right? And when we look at isotopes, the true difference between various isotopes is the number of electrons have to be the same. Okay, number of neutrons. Number of neutrons have to be different, right? So if you look at nitrogen 15, nitrogen 15 has 15. No. Nitrogen 15 has more neutrons than nitrogen 14. So if you look at the mass of it, nitrogen 15 is a heavier nitrogen, right? Heavier isotope. And what is DNA made up of? It is made up of nitrogenous bases, things that contain nitrogen. So this is where they make use of this concept. If I for example, have bacteria, okay, if I were to grow this bacteria in a petri dish containing just nitrogen 15, what I will end up getting is bacteria with DNA that contains only nitrogen 15. Okay, if you leave it long enough, all of this DNA if I leave this population to grow and grow and grow, most of the bacteria inside will end up having nitrogen 15 as their primary source of DNA. So if I were to kill all of these bacteria, extract out the DNA, take it for a spin in a centrifuge, what you'll find is that because this DNA is so heavy, it will sink all the way to the bottom of the test tube. Okay? Whereas, if I were to take bacteria and I grow it in a petri dish filled with the lighter form of DNA, lighter form of nitrogen, I will end up with bacteria containing lots and lots of DNA. They are very light because they're made out of nitrogen 14. If I were to kill all of these bacteria, extract out the DNA, take it for a spin, you'll find that these DNA molecules will be somewhere at the top of the test tube because they are lighter. They'll float to the top. But just with these two pieces of information, Mitosis and Stahl was able to discern which model was the actual model that is taking place within our cells. So let's begin with this generation over here. Assuming we have DNA, we have bacteria that is growing in, where should I put it in? Let me check. Let's grow our first generation of bacteria in nitrogen 15. We grow this original bacteria population in nitrogen 15, and we were to kill the bacteria and take it for a spin. Where would you expect the DNA to be located? Okay, generation 1. I take this bacterial colony, grow in nitrogen 15, take it for a spin. Where do you expect the DNA to be found? Passing to the bottom, right? Okay, so first generation we can expect definitely you'll find them all at the bottom of the test tube because they are very heavy. But let's take some of these bacteria, let them grow in the second generation. Okay? But this is where they got very smart. Rather than grow the second generation in a petri dish of nitrogen 15, let's grow it in a petri dish containing nitrogen 14 instead. Because you know what that means? It means that any new DNA that is formed will be nitrogen filled with nitrogen 14. It will be very light. I say again, any new DNA that is formed will be very light. The original that is still present will definitely still be heavy. If I were to take this population of bacteria and take it for a spin after killing the bacteria, where would you expect the bands to be? We will expect to find some on the bottom, right? We will also expect to find some DNA on the top. Yep. Let's look at a semi-conservative model. Assuming we start off again with growing bacteria in nitrogen 15, what you expect if I were to take the bacteria for a spin is that the DNA will sink to the bottom. Yeah, first generation. However, if the semi-conservative model of DNA replication were true, where do you think the DNA band will lie? If this model is true. Usually if you can understand this that means you understand this experiment. You're good to go. Okay? That Isabel is trying to whisper to me. Yeah. When you find this band of DNA, you end up finding them in the middle. And this is the part I want to check with you whether it makes sense to you. Why would you find the band of DNA in the middle? Okay, some students think that you'll find the DNA like this. You'll still find it like this. Okay? Okay, then I realize why some students think that you'll still find two bands. Okay, in reality, if I were to kill the bacteria, take them for a spin, what you will find is that these double-stranded DNA will stay together. They will not separate out when you spin them. Meaning to say, if I were to spin these two DNA molecules, they're not going to separate out like that. Cannot. Because we learned in JH3 that they are held together by hydrogen bonds across the whole way. So they will not break apart. Instead, what you'll find is that now I have DNA molecules that are half heavy, half light. And you will find them therefore in the middle. This was what Mitosis and Stahl predicted the results to be if they were to be conservative or if they were to be semi-conservative. And then they ran the experiment. And to their pleasant surprise, the experiment yielded this particular set of results. And from that, they realized that in fact DNA replicates in a semi-conservative way. They stay as half new, half old. Okay? What does this mean for you? The conservative model does not actually take place. Okay? What you watch in the video that I put on SLS, we are just showing you the two theories. But in practice, this is what shows when if you were to do the experiment. But it's very good proof that this DNA replicates in a semi-conservative way. How could we test your understanding of this particular experiment? We could ask you questions like, if I were to take this particular generation for conservative and go for one more generation, grow it one more generation of nitrogen 14, where do you expect to find the bands? If I were to take the semi-conservative model, take it for one more generation in nitrogen 14, where will you expect to find the bands? This is how you can stretch yourself a bit further. If we really want to mess with you and test your understanding, you may do this. Hey, I know Masterson and Stahl grow in nitrogen 14, but what if I mess with you? I grow in 15, then grow in 14, then accidentally put it back in nitrogen 15. Where will you find the band states? If you understand how this process works, then it's a little bit more of a puzzle for you to solve. Okay? So understand the concept, don't memorize this experiment. Because we will never ask you to redraw this experiment or to describe it. Instead, we will modify the experiment for you. So just a little puzzle for you to solve. Where do you expect to find the bands? If I take this generation, go to the third generation. Let me put the third generation here. So this is the strand, this is the strand over here. Okay, here's a clue. Whatever new DNA you produce needs to be heavy, right? So, as usual, each strand will serve as a template to synthesize new DNA. All the new DNA will be heavy. All the new DNA strands, I mean, will be heavy because we're growing N15. So how many bands will you see? Just two bands, right? One band will be in the bottom. One band will be in the middle. Okay? And this is how we can mess with you. Then maybe the next generation will go back N14. Then you tell me where you think you will go. Okay? So don't memorize this experiment. Instead, appreciate, understand how it works. That is good enough. Oh, okay. Good question. You need to ask, let's say if with every generation I get more and more heavy DNA, will it be a darker band? Is it? Okay, so two things could happen. It could look like it's a darker band over there if the next generation is more, or it could be a thicker band. Okay, so it could be a thicker band or it could be more DNA there. Okay. Questions for me? The last part of today, mitosis and meiosis. Let's get you all moving a little bit. I'll need the help of some people. Here on the whiteboard, I have some magnets. They are meant to represent chromosomes. What I will need your help with is we do a quick relay. Each one of you all, they are called up. You will come up and help to rearrange the chromosomes according to each phase of DNA of mitosis. Mitosis has 4 phases, right? So we have prophase, we have metaphase, we have anaphase, and we have telophase. What would the chromosomes look like, or how would they be arranged in each phase? So I'd like you to assume that this whiteboard over here is the cell. Okay? This whiteboard is the cytoplasm. Nucleus already broken down. Okay, nucleus already broken down. What I have here are chromosomes. First, I'm going to take away all this first and start with this. Here, I have a cell. This is the nucleus of the cell. Okay. Okay, I have a question for you. Let's say we look at the cell cycle. G1, S, G2, and then all the 4 phases. What would this nucleus look like, all these chromosomes inside, after S phase? Humans, how many chromosomes do we have? 36. Okay, I think we have 46. Humans have a total of 46 chromosomes. Okay, you have heard the word chromosomes in your learning. But what are chromosomes? Think of chromosomes as coiled up DNA. Okay? Chromosomes are nothing more than coiled up DNA. Whoa. Sorry, I got the whiteboard dust in my nose. Okay, chromosomes are coiled up DNA. DNA strands are double-stranded and they are actually really, really long. But if you leave them as it is, it's very hard to organize. So what the cells do is that they help to coil up this long DNA into more compact structures, and that's what we call chromosomes. So we have 46 chromosomes. But do you know that in reality, 23 of them come in pairs, meaning to say that for every chromosome you have inside your cell, you actually have a copy of it. The only pair that we have in our body that may not be the same are your sex chromosomes. For the ladies, it's still the same chromosomes. You have 2 X chromosomes. But for the men, you have 1 X chromosome and 1 Y chromosome. Your Y chromosome actually is much shorter. So actually guys, you have less DNA. Yeah? We actually have less of certain things. I don't know what this missing part results in. Maybe in our ability not to read this. Okay? So we actually have 23 pairs. Here on the whiteboard, I have a particular cell. How many chromosomes do you see here? Do a quick count. 6 chromosomes. Okay, so this particular cell has 6 chromosomes. How many pairs of chromosomes do you see? Okay, 3 pairs. So I'll let you understand that this particular cell I'm showing you over here is a different species. It is not human. Okay, this particular cell only has 6 chromosomes, 3 pairs. Okay? But it's easier to go quick on this small whiteboard. This is before S phase. I have a question for you. after S phase, how will this look different? Because we know that during S phase, DNA replicates, right? So after they replicate, what do you expect to see here? That's a good question. How many chromosomes will you expect to see after S phase? There are 6 chromosomes, right? So after S phase, so 6 chromosomes here. After S phase, it's a question. How many chromosomes will we expect to see? Okay, actually MCQ had an option, I give you 2 options. 6 or 12. How many chromosomes will you expect to see? 6 or 12? Okay, what makes sense is 12. What makes sense is 12. But in truth, it's still 6. Okay, why? Okay, so we know that by right, we are creating one more copy of each, right? So here, I replicate. Now I have 2. Here, I also replicate. Now I have 2. Here, I also replicate. Now I have 2. 1, 2, 4. Logically speaking, after S phase, I should have 12 chromosomes in total, correct? But actually, that's not the case, because our cell wants to be rather efficient in how we organize all these chromosomes. In reality, the replicated chromosomes are attached to each other, they join to each other at this center part called the centromere. So all replicated DNA, they will be temporarily attached to each other at a region called the centromere. We refer to one single unit like this. It's still a chromosome. Okay? So as long as you see individual units, they are chromosomes. We have a special name for this chromosome, it's a metaphase chromosome. Because you only start to see this over here, come to use from metaphase onwards. If you ever look into a cell and you see chromosomes in an X shape, understand that DNA replication has already taken place. How does this relate to what we just learned earlier on about semi-conservative replication? So earlier on we learned that one will be old, one will be new, one will be old, one will be new after you replicate the DNA, right? Okay. What I did not show you on the whiteboard, or rather what I could not show you, is that actually they will be joined together at a certain point in the middle. Then it will kind of be just one unit together. Okay? But that's what I could not show you. I say again, just now on the whiteboard, after DNA replication we end up with 101U, 101U like that. But how do they really exist in cells after DNA replication? They will join together. So they will join together, okay, if each one were to be one strand, each one were to be like that, what it will actually look like is that they will join together somewhere in the middle. In other words, if I were to look at this particular model here, each one of them is double-stranded. Okay? Why do we do this? Or why do cells do this? You'll see that it is really for convenience, efficiency. It makes it very easy for us to separate things later on. Yes, Shen? Oh, okay. Okay, I understand. Hold on, I'm repeating. How do I respond to your answer? I think what the experiment I showed earlier on, what I did not account for is where all these are present. So just now, right, although there were, okay, what I did not account for or show you was mitosis. So in reality, you will not have a cell where you have a situation where you have 1, 2, 3, 4 different things, 4 of the same things together, and for you to figure out how to join. Because mitosis will have occurred for them to separate out evenly. Oh, which one are the cells? I would say earlier on what I should have done was to show you that there are actually 2 cells. Yeah? And then the 2 cells therefore ended up having, but on the screen you saw 4 different DNA molecules, right? But actually the 4 different DNA molecules, what I should have shown you is that they are present in 2 cells. Yeah. Okay? Yes?
Is this during prophase?
Oh.
Or just during phases?
Okay, so now you're asking, when will you see this, is it?
Yeah, because I thought like they will condense only in prophase.
Okay, so you're right, it will condense only in prophase. So is your question, when will you see this condensed form? Okay? Okay. You will see this condensed form from prophase onwards. What will you see in G2 phase, therefore? What you are seeing here on the screen is from here onwards, from prophase onwards. But what will you see in G2 phase? In G2 phase, you see something like that. So let's rewind a little bit. If you rewind to G2, what you will see is that this over here will be uncoiled double-stranded DNA and uncoiled double-stranded DNA, but still joined together at a certain point, but uncoiled. The moment it transits into prophase is when you see it start to compress to form this more dense version. It's easier to move around. Okay, so when will you actually see these compressed star X-shaped sausages? Kind of from prophase onwards. Prophase is when it starts to really form. By the end of prophase, you see it. Metaphase, you see it most clearly. And that's why in some sources they call this metaphase chromosomes. Because you really see it from metaphase onwards. Okay, so now you do a very quick revision of where you expect all of these fellas to move within the cell as the phases progress by. Let's start off first with prophase. Okay, so I have these 6 metaphase chromosomes here. Okay, in prophase, how will you expect these 6 chromosomes to be arranged within a cell? If you are ready, now I'll call someone. You can come up, you can rearrange it to your liking. How will you expect the chromosomes to be arranged, let's say at the end of prophase? End of prophase. What I have still left on the whiteboard is the nucleus here. Yeah, so end of prophase. How will you expect these 6 chromosomes to be rearranged? Will they be arranged? Okay, prophase. Okay, whatever I call up to you what you want to do to these 6 chromosomes. How do you think you'll be arranged at the end of prophase? Shanice? Or you and Madhu can go on together.
It's attached to the pseudofibers.
Oh, okay. Do you come out or you come out? Why you can draw? Sorry, this is lah. Sure. Okay. Oh, okay. I see. Okay. Thank you. Okay, thanks, Shanice. So, okay, Shanice, what are these circles you're drawing? Okay, what are those called? It starts with a C. Let's play hangman. Hey, correct, okay. Sand. Okay, it's not a center summit, it's a sand. Okay, okay. So, central. Can you look at your textbook? Can you see the word? Can you find the word? Okay, Shanice, go and sit down. Thank you. Thanks, Shanice. Can you find the word central? Yeah. Okay. Okay. So, if you were to look at your textbook, centrals they tend to draw it with two parts. There's like they tend to draw as one rectangle and another rectangle like that. Okay. Then Shanice also drew lots of fibers coming out. Thank her for that, and with Manu's help. So these are called. Okay, so you use the word spindle fibers, right? Okay, that's fine. Spindle fibers. Indeed, they are fibers. What are spindle fibers made up of? They are made up of. Do you learn this word? Microtubules. Okay, micro means microscopic. Tubules means tubes. So spindle fibers are just made up of many microscopic tubes. Thank you, Shanice, for drawing all this, because indeed, during prophase, that's what's really happening. The centrioles act kind of like fishermen. And what these fishermen are doing is they are throwing out the line. They are throwing out the line, which are the spindle fibers, the microtubules, and slowly reeling in the chromosomes. Okay? I say again. So imagine there's a fisherman here. He threw out a line. Right? And then he managed to hook on to this chromosome here. This fisherman over here threw out the line. Right? And then managed to hook on to this chromosome here. In prophase, that's what a lot of these centrioles are doing. We are trying to attach the microtubules, the spindle fibers, to the chromosomes. But why so? And what happens if the fishermen on both ends attach to the same chromosome? So let's look at this one up. This fisherman attached here. Okay, this fisherman also accidentally attached, or actually surely he will attach one microtubule here too. Now they are both pulling on both ends. Kind of like a tug of war now. Yeah? But actually this is a very important thing. Because as a tug of war, these two fishermen pull on both ends and shorten the line and they slowly reeling in. Something will happen in the next phase. Let's go to the next phase. What will it look like now in metaphase? Yes? Oh, the centriole is where the mitotic spindle comes out from. Yeah. So that's why I like to see the centrioles as like fishermen. They're just fishermen sitting there with tons of fishing rods and then they just, right, they throw out all of their fishing lines. So next person, what would metaphase look like? Supposing all of these centrioles have managed to attach themselves from both ends to each chromosome. Wow, okay. So all of them attached to both ends. Okay, it's a mess already. What will it look like metaphase? If both centrioles shorten, they are microtubules. What would metaphase look like? When I invite you up? Okay, free for all. Okay, erase everything here. Just like you to primarily focus on where all these chromosomes will be after the centrioles do their thing. And don't know if my whiteboard got space for you to do what you want to do. Ah, okay, okay. You need more space, is it?
I think this is fine.
Okay. This one, eh? Okay, I'll put a bit higher. Try like that. What would the spindle fibers and the microtubules look like if we were to overlay them here? Okay, thank you. What happens if both centrioles, the tension is equal? They pull the line in until both sides, the tension is exactly the same. The tension is exactly the same on both sides. Then you will observe the chromosomes kind of lined up in the middle, or what we call the equator of the cell. I noticed, I don't know if you all saw this, Yuan Yuan originally drew the microtubule to attach here. Then she erased it away, and then she redrew it to attach here. I wonder if it makes a difference where you attach it. Because how you attach it makes a difference. Notice that Yuan Yuan deliberately attaches the microtubule to one strand here, one DNA molecule here, attaches to one DNA molecule here. It has metaphase, because in the next phase, this is where it comes in very useful. What would the next phase look like? Now, if each centriole gives one last tug and pull, as hard as possible, then something will happen next. It won't snap, the microtubules will not snap. What will happen next? Okay, I will play the story out. If they were to do one final pull, all of these chromosomes, all of these metaphase chromosomes will finally split apart. And they start to pull it to opposite ends. Yeah? Into their new homes, new nucleus. And do you appreciate why it is important to stick them together at the very beginning? Because when we stick them together, it makes it easier for us to organize them later on into two nuclei and make sure that each nuclei has exactly one copy. Okay? So this is the beauty of cell division. Nuclear division. I have a question for you now. On the whiteboard, how many chromosomes do you see now? Okay, now it's 12. Okay, so always remember, chromosomes refer to one entity. So now I have 12 chromosomes on the screen. Okay, let's play a game. How many chromosomes do I have on the screen right now? Wait, is it 11? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Okay, yeah, okay? Okay, if I'm going to join these two, how many chromosomes do you see on the whiteboard? 10. Okay, so chromosomes refer to separate individual entities. They must be together or apart. So here I have 12 chromosomes now. No longer called metaphase chromosomes, these are daughter chromosomes. But from here onwards, a lot of things make sense. They will then go into their individual nucleus, will reform nucleus. This is telophase. And then the cell will divide into two big cells. Each cell now will have the same number of chromosomes it started off with. And what was it? 6 chromosomes, 3 pairs. Yeah? And that was our starting point. That's mitosis. The skill therefore now is, this is all in theory, but if you were to see real images under the microscope, what would it look like? If you look at your textbook, what your textbook is showing you is not what you will see under the microscope. It's showing you even more high-res image. I think what would help is if you look at practice makes progress. Wow, let's look at these four. How do you tell which phase of mitosis these four cells are in? Okay, what has helped me as a student is that actually if you go online, mitosis, micrograph, phases. I found this particular image that I quite like. That shows you, number one, on the right hand side, the theory of it, and on the left hand side, what you may see under the microscope. Okay, let's look at what are the key features to look out for to help you identify. We start off with interphase. Okay, in interphase, we know that the nucleus is still present. DNA not yet condensed. So actually what you will see in real life is this. You will see a cell with the nucleus kind of in a circular shape because the nuclear membrane hasn't broken down. You will also see lots of like dots. These dots represent uncoiled DNA. Okay, these dots represent uncoiled DNA. What does coiled DNA look like under the microscope? It looks more like this, and this, and this, and this, and this. Coiled DNA look like fat worms. Uncoiled DNA has no structure. They just look like dots. So that's how I differentiate the phases. So interphase, a defining thing you will see is that it's still in a circle and lots of dots. How do I identify prophase? Okay, this is early prophase, this is late prophase. But in general, how do I identify prophase? I identify it by, number one, I see lots of fat worms. Yeah? Fat worms that have no particular orientation, no organization. What do I mean by organization? Look at here onwards, notice that they are organized in a certain way. But for prophase, not much organization. In fact, if they are organized, it is kind of in a circular way because the nucleus just broke down and they are starting to spread apart. How do I identify metaphase? Notice that metaphase looks nothing like what you will see in your textbook if you were to see it in real life. So I need you to start to get used to this particular image over here. Metaphase actually looks kind of like that. The chromosomes almost look like they are arranged in this fashion. If you were to look into a nucleus, they kind of look like they're all pointing inwards. Although you cannot see the chromosomes clearly, can you see that they're kind of all V-shaped pointing towards the center? That's how I identify metaphase in real life. How do I identify anaphase in real life? Anaphase in real life, you kind of observe that the V shape is pointing away. Can you see that it's kind of pointing away? That's how I identify anaphase. Last but not least is telophase. Telophase, you notice that if you recall, in telophase, DNA starts to decondense. Therefore, you don't really see the thick worms as clearly as possible. They start to become a mess. They're starting to uncoil. If you can find this image, you can go and paste it into your notes. I think it will be quite helpful for yourself. I searched up this particular set of words: mitosis, micrograph, phases. So this is a skill to pick up. You must be able to identify the phases of mitosis from microscope images. If I were to show you this particular question again, although not quite similar to what we saw earlier on, can you roughly tell which phase is which? What did the question ask us? It asked you to order it. Name D. Okay. Which phase do you think is which? Personally, I find it quite hard to identify some phases, so I kind of go by a rule it out thing. In this phase, I feel that I'm more confident in. Because out of all the phases, this looks like one of the phases. What phase do you think this is? Okay, I think it's closest to metaphase, so this seems like the most obvious. B is actually quite hard to identify, right? Can you tell if it's prophase or if it's interphase? Bit hard to tell. But what I can rule out is it's not going to be metaphase because I don't see it organized well. I can rule out it's anaphase because it doesn't look like it's being pulled apart. So I'm going to make a wild guess that it's either pro or meta. Hey, no, sorry, pro or interphase. But I think it may be more prophase because you don't see the nucleus anymore. So that is my next best guess. Okay, let's look at these two. They look kind of similar, right? What would be the biggest difference between these two then? Sorry? The separation. This one kind of looks more separated than this one. So, by logic, I think this definitely needs to happen very late into the phases. So, although I may not be right, I think our best guess may be telophase. It has to be quite late. But then only left with a few phases left to play with, right? So this one, although it may not look the best, it may be anaphase. Because it looks like it's going to two ends. Or it could be very late telophase. But you notice even the teacher don't know. That's why we ask you to identify stage D. Yeah? We ask you to identify the most obvious one. Help us, help you. Okay? But what we could still ask you to do is to sequence it, even though you do not know the stages exactly. Okay, so these are some examples of questions you could encounter that ask you to identify the various stages. You'll get one more term review, I think the final one, and then you can practice more identification of phases. So today we just revised mitosis. Meiosis, if you realize it's just the same process, but happened twice. Okay? Okay, so tomorrow, Tomorrow I think I'll have a bit of time. I'll go a little bit on meiosis, and then we'll start reproduction already. For reproduction, we'll start off slowly. We'll start off with learning about the parts of our own body. It's not awkward until we make it awkward. Okay, last 2 minutes. You just consolidate, do whatever you want. Tidy up your notes. What's next, Marita? Your timetable changed significantly? No, yes. Jump what? Is that good? You know the orange I always carry around in my container? Yeah, the orange is Chinese New Year. Well, over the holidays, the aircon got switched off. When I came back today, right, it's just black, flattened mess. All the content of inside rot and ooze out into my whole body. Yeah, go. Yes, you can. Okay, can you stop my recording?